Good morning, students. Today we are going to study about T4 phage. You know what are phages? Bacteriophages. They are bacteria eating viruses are called as bacteriophages. And T4 phage is an example for bacteriophage. Now you are seeing the diagram of T4 phage. It has head, neck, tail region and tail fibers. So when you take the head region, it is icosahedral in shape, and next to the head region you can see a small collar portion, and behind that collar portion you can see tail, and the tail is in cylindrical extended portion. It has a contractile sheet covered by the tail is covered by contractile sheet, and at the bottom it has a basal plate, and in the basal plate you can see 6 tail fibers. So this is the structure of T4 phage. Coming to the reproduction of T4 phage, it follows two types of reproduction: lytic phase and lysogenic phase. And most of the phages follow lytic cycle. And in lytic cycle, first we'll see about lytic cycle. So lytic cycle comprises four stages or four phases. First one is adsorption, penetration, synthesis, assembly and maturation, then release of the new phages. Now see one by one. First step is adsorption. In adsorption, attachment sites on the bacteriophage adsorbs to receptor sites on the host bacteria. So you can see, so the virus is being adsorbed, attached to the bacterial cell, surface of the bacterial cell, the cell wall. They adsorb to the cell wall. Some may adsorb to flagella or pili, etc. Here, specific strains of bacteriophages can only adsorb to specific strains of host bacteria, and this is known as viral specificity. And the next step is penetration. So, first step is adsorption. After adsorption, there is penetration. In the case of bacteriophages that adsorb to bacterial cell wall, a bacteriophage enzyme make a hole in the bacterial cell wall, and it injects the viral genome is injected into the bacterial cytoplasm, and this process is called as penetration. In some bacteriophages accomplish this penetration by contracting a sheath which drives a hollow tube into the bacterium, and the genome is injected into the bacterial cytoplasm. The third step is replication. After injecting the viral genome into the bacterial cell, next step is replication of the viral genome. Here, enzymes coded by the bacteriophage genome shut down the bacterium macromolecules, that is the protein synthesis, RNA and DNA synthesis of the bacterial cell is shut down. Then bacteriophage replicates its genome and uses the bacteria's metabolic machinery to synthesize bacteriophage enzymes and bacteriophage structure components. The fourth step is maturation. After the synthesis of viral genome, its structures, etc., the fourth step is maturation. Here, the different parts the different parts of the virus synthesized will get together or assembled around the viral genome. This process is called as maturation or assembly. Then, after the formation of the new viral particles, there is breaking of the bacterial cell wall and the release of new bacteriophages takes place, and this step is called as release phase, and this is the final step in lytic phase, life cycle of reproduction. Coming to the next lysogenic cycle, here the bacterial cell wall is not lysed, and so only it is called as lysogenic cycle. Here the first step, the phage gets attached to the host bacterial cell. It injects its genome into the bacterial cell. Then after the entry of the prophage, that is the viral genome, it gets attached or integrated with the bacterial genome. You can see here the viral genome is getting attached or integrated with the bacterial genome. Then here the viral genome divides along with the bacterial genome, and it goes on like that. And after some time, what happens during favorable condition, once again the viral genome detaches from the bacterial genome. It undergoes replication. The viral DNA undergoes replication, and other viral structural parts get synthesized, and there is assemblage of the virus, daughter viruses. And after the formation of the daughter virus inside the bacterial cell wall ruptures, and the daughter or new bacteriophages are released out of the live bacterial cells. In It also includes 5 steps. The first one is attachment. Here, the bacteriophage identifies and attaches to the bacterial cell. Then next step is penetration. Then after penetration of the viral genome into the bacterial cell, the integration takes place. Then latency period. Then during induction, once again what happens? The viral DNA is detaching from the bacterial genome, and it undergoes synthesis, and new daughter viruses are formed, and after lysis of the bacterial cells they are liberated outside. And this is about the lysogenic cycle of bacteriophages. So I think you might have understood the two different types of reproduction, that is lytic cycle and lysogenic cycle of T450. Thank you for listening.